Welcome to the video tutorial of pluriBead Cascade. pluriBead Cascade allows you multi-target separation from the same sample material. That means isolating two cell types at the same time. To get started, you need the S pluriBead kit, for example for CD4, and the M pluriBead kit, for example for CD8. Here are the necessary items in detail. You need the S pluriBead for your first target, a compatible M pluriBead for your second target, the pluristrainer S and the M pluristrainer, connector rings, a funnel, detaching buffer, your sample material, and washing buffer. Additionally, you will find the manual. To start your experiments, add the two different pluriBeads to your sample material. M pluriBead means medium size and is labeled with a blue cap. S pluriBead means small size and is labeled white. Before adding the pluriBeads, resuspend them well on a vortex. After closing your incubation tube, premix the sample material on a vortex or by tilting overhead a few times. Incubate the sample on a pluriplix or a wiping rolling mixer for 5 to 30 minutes at room temperature and a speed of approximately 10 to 15 rpm. During the incubation, you can start the next step by assembling the pluristrainer Cascade. To do so, take out the stereo S pluristrainer from the blister and put it on a fresh 50 ml centrifuge tube. Then attach the M pluristrainer on top of the S pluristrainer. Make sure that the S pluristrainer is on the bottom of the Cascade. Now you can attach the funnel to the Cascade. Before pouring the sample material over the Cascade, equilibrate the pluristrainer with 2 ml of wash buffer. When your incubation is finished, pour your sample into the funnel. Your sample will run through the Cascade by normal gravity. Wash your incubation tube twice with wash buffer. In the next step, you wash the wall of the funnel with wash buffer. Make sure that the buffer runs through the Cascade completely before adding the next washing buffer. After washing, discard the funnel. Now wash the Cascade sufficiently with wash buffer. There are two goals to achieve. First, washing off the unbound cells, and second, separating the M pluriBead from the S pluriBead. Wash until you see no red, and then wash 3 times more. Now you can disassemble the Cascade by dividing the two pluristrainers. Put the upper pluristrainer on a new 50 ml centrifuge tube. Wash both pluristrainers again. To prepare the detaching step, attach the green connector rings tightly and even to a fresh and sterile 50 ml centrifuge tube. Now press tight the pluristrainer to the connector ring. Make sure that the pluristrainer is not tilted and the lure lock is closed. Otherwise the detaching buffer will drip through. For detaching, add first 1 ml of washing buffer, and then 1 ml of detaching buffer. Detach for 10 minutes. During the detaching, resuspend the beads by swirling, all 2 minutes. Do not pipette at this step. After 10 minutes, add another milliliter of wash buffer and pipette the detaching solution 10 times in a circular way. This step is more simple by holding the tube angled in your hand. Make sure not to hit the sieve during the pipetting. Now you can open the lure lock. The detached cells will run through and the pluribeads will stay on top. Wash the pluri strainer with washing buffer or your cell culture media, 5 times 2 ml. Discard the pluri strainer and connectors. For spinning down the cells, for your further experiments, we suggest to use a 50 ml centrifuge tube. That will lower your cell loss by approximately 30%. As you can see, with pluriBead Cascade, it's very easy to separate 2 targets at once. It saves you time and sample material. We hope this tutorial was helpful for you. In case you have any questions, please contact us under support@pluriselect.com. Thank you for watching.